a particular gene into a target cell. Um, and expression vectors must have expression signals such as strong promoter, a strong terminal codon, adjustment of distance between the promoter and a clone genes, and insertion of a transcription termination sequence and a portable translation initiation sequence. Expression vectors are used for molecular biology techniques such as site-directed mutagenesis. Basically, in expression vectors, when inside the cells, the protein that is encoded by these gene is produced by the cellular transcription and translation machinery ribosomal complexes. The plasmid is frequently engineered to contain a regulatory sequence that act as an enhancer and promoter regions and lead to efficient transcription of the gene carried on the expression vector. The goal of the well-designed expression vector is the production of large amount of stable messenger RNA and in extension proteins. Expression vectors are the basic tool for the biotechnology and the production of proteins such as insulin, which is important for treatment of diabetes. Expression vectors produce protein through the transcription of vectors insert followed by the translation of mRNA produced. They are therefore require more component than the simpler transcription only vectors. Expression in the different host organisms would require different elements, although they share similar requirement. For example, a promoter for initiation of transcription, a ribosomal binding site for translation initiation and termination signals. So in prokaryote expression vectors, um, as we can see in the diagram, there is a clone gene and a AMP, which is ampicillin resistant gene of PBR322 resistant plasmid. So sorry. Yes. So promoter commonly used inducible promoters are prokaryote derived from the lacoperon and T7 promoters. A stronger promoter, TRP or tryptophan operon and TAC promoters, a hybrid correction of both the TRP and lacoperon promoters. Ribosome binding site RBS follows the promoter and promoter efficient translation of the protein of interest. Translation initiation site that is Shine-Dalgarno sequence enclosed in the RBS is 8 base pairs upstream of the AUG start codon. The basic requirement of any expression vector expression system is a promoter cloning site. Next to it is a transcriptional terminator. Origin suitable for the replication initiation in a particular bacteria or any host. A selection marker gene such as antibiotic resistant gene. Regulators element Shine-Dalgarno sequence. These are basically the parts of expression vectors or component. So the example of expression vector is PET expression vector. So expression vector is vector in an which is used for the expression of recombinant protein in E. coli. This vector system utilizes the T7 promoter system for the control gene expression. It is also used as a bacterial recombinant protein vectors. And the size of this vector is 5700 base pairs. It has T7 promoter adjacent to LAC operator. Next to it is a sequence called Shine-Dalgarno sequence, which is basically a ribosomal binding site in a bacterial or a archaeal messenger RNA. Adjacent to the Shine-Dalgarno sequence, there are the few cloning sites such as NDE1, which is type 2 restriction enzyme, and NHE1 and BAMH, which is type 2 restriction enzyme, restriction endonucleases enzyme, and this is for recognition of short sequence and cleaving them at target site, and at the end of which there is a T7 fast transcriptional terminator is found. For the expression of these genes, the bacterial cells should provide T7 RNA polymerase which is under the control of LAC-Z operator or promoter. The repressor produced by the bacterial LAC-IQ can be regulated by IPTG, and this is a diagram of PET expression vector. These are references. Thank you. Sir, recombinant plasmid is basically um a plasmid which is used for uh, modification of DNA, specific DNA sequence. Sir, through, sir, I don't know. Sir, in basically expression vectors, they just they just act as a plasmid, and they produce a sequence which can be used as a mutating agent in cell or a DNA. No, no, sir. Sir, they often they act as a plasmid, um, which can use for mutation. सर एक्सप्रेशन वेक्टर बेसिकली लार्ज अमाउंट ऑफ स्टेबल आरएनए प्रोड्यूस करता है जो कि आगे जाके प्रोटीन प्रोड्यूस कर सकता है सो क्योंकि वो एज कंपेयर टू बाकी वेक्टर से ज्यादा स्टेबल होगा इसके लिए उसको यूज कर सकते हैं सर थ्रू पीसीआर ओके सर सर
हेलो सर
अपनी
नो सर
लगा लिया किसने किसने देखा ये
नो सर
यस
Sir, should I present? Okay. Sir, is it visible? So, good evening, everyone. My topic is methods of transformation of host cell, roll number 227313. So, what is transformation? Transformation is the process that allows bacteria to uptake free DNA from their surrounding environment. The transformation was discovered by Frederick Griffith in 1928 by experiment on Streptococcus pneumonia. Bacteria that undergo transformation are called transformants. So examples of transformants. There are basically two types of transformants, natural transformants and artificial transformants. So natural transformants, which comes under gram-negative bacteria are Haemophilus influenza, Heliobacter pylori. Natural transformable gram-positive bacteria are Bacillus subtilis, Streptococcus pneumonia. Natural transformable acid-fast bacteria are Mycobacterium streptomyces, and artificially transformable bacteria are Escherichia coli and Salmonella typhi. So, when do bacteria cells take up DNA? So, bacterial cells take up DNA is called competent cell. So what is meant by competent? Competent cell means the cell which is ready to uptake foreign DNA. Competency is induced when the number of cells in the given volume is increased by a certain level. In gram-positive spore-forming bacteria, Bacillus subtilis, establishment of a natural competency occurs when the cell reaches the high density towards their exponential growth. So here is the diagram uh, which represents the machinery of natural transformative cell, Bacillus subtilis. So transformation, as we know, transformation is the process that allows the bacteria to uptake free DNA from the surrounding. So Bacillus subtilis, which has similar mechanism for DNA binding and transportation proteins that bind DNA and encode by some COM genes. Uh, these DNA binding receptors are assembled and placed on the cell surface. The DNA binding receptors plug the dsDNA molecule from the pilus. So we can see in the diagram, the pili of Bacillus subtilis bind the DNA on the cell surface and the cell type 4 pili. Then the DNA binding receptors is the COM-EA protein which bind the double strand DNA but has no sequence specifically. So in the third step, the COM-G protein makes the COM-EA receptor accessible to DNA. Once the DNA is irreversibly bound by the COM-EA receptor and the DNA enter the nucleus degrades one of the DNA strand. The COM-I protein is a membrane bound nucleus responsible for this. So the remaining DNA strand is transported into the cell with the help of the receptor protein COM-EA and COM-EC. Inside the cell, a single strand of DNA is protected by a COM encoded DNA binding protein. This DNA protein complex can undergo homologous recombination with the chromosome and chromosome of recipient cell if homologly exists and if the cell are REA plus. So in this way, Bacillus subtilis goes under natural competency. So where does this free DNA comes from? A bacterial cell might have a unfortunate encounter with lytic bacteriophage. This liberating bacterial chromosome DNA into the environment when the phage lyses the bacterial cell. Alternatively, a bacterial cell might become an unwilling donor for a free DNA if is where the encounter hostile environment conditions that cause cell lysis, such as inappropriate salt of pH concentration, harmful chemicals, or very aggressive immune system. So now artificial transformation. So how does artificial transformation takes place? Many bacterial species, especially those exploited for biotechnological purpose, are not naturally transformable. This is because they do not contain genes that encode product needed in the uptake of a transport DNA. Many bacteria can be made component by the exposure to a divalent or multivalent cation such as calcium chloride, magnesium chloride. This take place at very low temperature. Bacteria treated with cation, which has positive charge, and bacteria is slightly negative. That's why stand besides calcium ions. When current is removed, lipid barrier comes in normal position. This is how artificial transformation is done. Thank you. So what? Hello. So don't. Sir, calcium chloride increase the permeability of the bacterium membrane, making them chemically component. And role of com I. So one second. I know the role of com I. Uh, uh, Actually, com I is uh, uh,
सर लास्ट में भी तो चलेगा ना।
Yes, sir. हाँ सर. सर थोड्या वेळाने देते. East artificial chromosomes. My topic name is uh, introduction. Each artificial chromosome is a human engineered DNA molecule used to clone DNA sequence in each cell. Segments of an organism DNA up to 1 million base pair in length can be interested in each artificial chromosome. Each artificial chromosomes are plasmid shuttle vector capable of replicating and being selected in common bacterial hosts has Escherichia coli as well as in the budding yeast Saccharomyces cerevisiae. They are of relative small uh, size and of circular form when they are amplified or multiplied in E coil but are rendered linear and of
very large size, therefore survive hundreds of kilobase when introduced as cloning vector in yeast. The yeast artificial chromosomes with their interested DNA are then taken by yeast cells. As the yeast cells grow and divided, they amplify the egg DNA, which can be isolated and used for DNA mapping and switching. The latter process involves cleavage at strategically located sites by two restriction enzymes which break them into linear DNA arms. These are subsequently ligated with the appropriate DNA insert before transformation into respect yeast cells. In this linear form, this specialized vector contains all three cis-acting structural elements essential for behaving like natural yeast chromosomes and autonomously reflecting sequence necessary for reflecting a centromere centromere for segregation at cell division and two telomeres for maintenance. Each artificial chromosomes survival advantages over other large capacity vectors. These include accom- accommodation of DNA segments, thousands of kilobase in size, and stable maintenance of electric clone eukaryotic DNA due to the capability with the yeast replication machinery. Yeast artificial chromosomes plasmids. Many different yeast artificial chromosomes plasmid exist, such as yeast artificial chromosomes plasmids 3 and yeast artificial chromosomes 4 plasmids. The basic structural features of yeast artificial chromosomes were developed from the yeast centromere shuttle plasmid series. These are composed of double standard circular DNA sequence carrying the B-lactamase gene and the bacterial PMBI organ of reflection, thus uh, conferring resistance to amplification and the ability to reflect in bacteria reflectively. They also include yeast ARSI with its isolated CAN4 DNA sequence as well as the URA3 selectable marker on this basic stafful plasmid. The yeast is cloned flanked by a telomere-like DNA sequence that are adjacent to regions sites for the uh, restriction enzymes. Uh, most of the yeast artificial chromosomes also contain the cloning sites in the middle of the superior of an allele of a tyrosine transfer RNA gene. This is the circular map of plasmid vector, plasmid yeast artificial chromosomes, plasmids 3. Biological features of yeast artificial chromosomes. First, large DNA is ligated between two arms. Each arms end with a yeast telomere so that the product can be stabilized in the yeast cells. Interestingly, large yeast artificial chromosomes are more stable than store ones, which favor cloning of large stretch of DNA. One arm contains an autonomous replication sequence, a centromere, and a selectable marker. The other arm contains a second selectable marker. Third, insertion of DNA into the cloning site inactivates a mutant expressed in the vector DNA and red yeast clonings appear. For transferred are identified as those red clonings which grow in a yeast cells that is mutant of TRPL and URU3. This ensure that the cells has received an artificial chromosomes with both telomere of complementation of the two mutation and the artificial chromosomes contain insert DNA. Consort of yeast by homologous recombination. The potential to use yeast artificial cloning technology has been enhanced by the ability to use homologous recombination for manipulating exogenous DNA in the yeast host in recombinationally target yeast artificial cloning. Yeast artificial chromosomes are assembled in vivo by recombination and not by ligation or in vitro. Recom- re- Recombination takes place between a target segment of the exogenous DNA and the yeast artificial chromosomes vector that contains sequence homologous to this target's transformation of the two yeast artificial chromosomes vector arms and the exogenous segments flanked by the target segments followed by recombination result in the formation of the described stable yeast artificial chromosomes. This is a diagram recombinational target cloning with yeast artificial vectors. Uh, Application of yeast artificial chromosomes withholding of DNA inserted thousands of kilobase in size and preservation of cloned eukaryotic DNA due to the capability with the yeast reflection mechanisms. Second, the artificial chromosomes correspond to the top tools for the learning and eukaryotic genomes and for mobilization of your genetics elements among bacteria and eukaryotes. Chromosomes translated it can be studied by means of disposable yeast artificial chromosomes that do not contain genetic information necessary for cell functions. Another major application of yeast artificial chromosomes is in the study of regulation of the gene expression by cis-acting controlling DNA elements that are present either upstream or downstream of large eukaryotic genes after the transfer of the yeast artificial chromosomes from yeast to mammalian cells. Artificial chromosomes vector have made easier the mapping and switching of complex genomes at a progressively more rapid rate. Conventional bacterial cloning agents have remained significant for the learning of other short clones, but for the cloning of very hang DNA species, artificial chromosomes have totally replaced pure bacteria systems, counting the label phage-based chromosome vector references. And thank you.
As
Sir,
Yes, sir. Sir, it's visible. So, good morning, sir. My name is Vaishali Yadav and roll number is 227335 and my topic is Cosmid. So, what is cosmid? It is a type of hybrid plasmid. It contains lambda fast cos sequence, or it is cosmid is made up of cos site plus plasmid. They contain cos genes of the bacteriophages, lambda bacteriophages. Genomic size of cosmid is about 30 to 52 kb, or normally 45. If they have a suitable origin of replication, they then they can replicate as a plasmid within the host cell. For example, SV40 ori and call even ORI. It also contains selectable marker such as ampicillin resistance gene. Collins and Holmes in 1978 was first to describe cosmid. And characteristics of cosmid: cosmid are plasmid that contain the lambda cause or cohesive ends site that are the lambda sequence required for the packing of DNA into a lambda phase particles. Cosmid also contain a plasmid origin of DNA replication, at least one selectable marker, and useful restriction endonucleoside. Features and uses of cosmid. Cosmid vector are designed to clone large fragment of DNA and to grow their DNA as a virus or as a plasmid. Cosmid vector are used in a homologous recombination between two different plasmid in the same cell and grown in a both bacteria and animal cell. Cosmid vector along with a lambda vector are used as a standard vector for the cloning or genomic DNA. Some examples of cosmid. The classic example of cosmid vector is C2RB, which carry an C2BR है, which carry an origin of replication and a cloning site and has a antibiotic resistant genes. And commonly used vector is PHB79 and supercos1. Here the diagram of supercos1. Uh, um, how the cloning of origin DNA in cosmid vector involve the following step. So first of foreign DNA, which is cut by a restriction enzyme, and also the cosmid vector is also cut by the restriction enzyme. The circular cosmid is form in linear form, and then foreign DNA and cosmid, which is a uh, like the means मतलब join करते by DNA ligase and it forms linear. Then the huh, the form it is packed in a phase or virus head, and then by the transduction it is introduced in E. coli as a plasmid. Here the steps. And the example of cosmid is supercos1. The property of supercos1 is a novel 7.9 kb cosmid vector that contain a bacteriophages promoter sequence flanking a unique cloning site. Cloning capacity of supercos1 is 30 to 42 kb for generating cosmid library, ideal for high resolution restriction mapping and rapid chromosomal walking. This structure allows rapid synthesis of walking probes specific for the stream end of insert DNA. The supercos1 vector is also engineered to contain genes for the amplification and expression of cosmid clone in eukaryote cell. Here the advantage and disadvantage of cosmid. One of the advantages of cosmid for the construction genomic library for organism with a large genome in that they have a cloning capacity about twice that of lambda vector. And disadvantage is that some cosmid clones are unstable on a propagation in E. coli due to the high copy number plasmid replication system. Here the references. Thank you. Cosmid is much more better than plasmid. Uh, रीजन उस जो कॉस्मिड है मतलब उसमें 1 और 2 कोस साइड होते हैं. कोस साइड और उसमें मल्टीपल ये सिलेक्टेबल मार्कर भी होते हैं और means recombinant mean नहीं सर तीन ही मुझे एग्जांपल मिले मैंने गूगल से सर्च किया था जो मैंने बताया था C2BR जो कि क्लासिकल यूज है और दो कॉमन है PHV79 और सुपर कोज 1 थैंक यू सर
Yes, sir. 1 minute only. सर मैंने आपको प्रेजेंटेशन शेयर किया है.
Yes, ma'am. दिख रहा है.
Yes, sir. My name is Rutuja Jagan Yadav and my presentation topic is shuttle vector. A vector that can replicate in more than one host organisms or two different cell types is known as shuttle vector. Example, a prokaryotic cell and a eukaryotic cell. An example is a yeast shuttle vector that can propagate within the cells of E. coli and yeast. Another is the adenovirus shuttle vector that can propagate in E. coli and mammal cells. Shuttle vectors are that carry origins of replication from two different hosts, which enables them to shuttle between the two hosts. These vectors contain DNA plasmids that can usually replicate in both mammalian cells as well as bacterial cells. Shuttle vectors function as hybrid vectors containing DNA sequences from bacterial plasmids and mammalian viruses. The vectors contain three functional DNA sequences involved in the cloning process: a viral replication origin, a bacterial replication origin, and a drug resistant gene. The presence, the presence of different replication sites and repair sequences enable the recovery and maintenance of these vectors in bacterial cells. There are there are three different vectors depending on the type of replication system utilized by the vectors. First is transitioning uh, replicating shuttle vector that need to be recognized by large T antigen in order to replicate in human cells. Second is episomal shuttle vectors work to establish cell lines that can replicate permanently in the form of plasmid DNA containing the DNA insert. Third one is integrate shuttle vector, which undergoes replication only after fusion with particular cell types for gene expression. This is the diagram of shuttle vector. Uh, sir, again. An example of shuttle vector with two different replicants for E. coli and cyanobacteria is PSCR119 that contains the E. coli, PMB1 replicon, and the PDC1 plasmid replicon for Nostoc MAC, which enables throttling of DNA contracts between E. coli and Nostoc. Advantages and disadvantages. First is the advantages. The main advantages of shuttle vector is that they can be manipulated and grown in E. coli and then used in a system which is more difficult or slower to use. And we can use it for both expression purpose and cloning purpose. Disadvantages: carrying capacity is low, not that is not longer than to 20 kb. Thank you. Hello. Uh, sir, don't know. Recombinant vector. The recom. Sir, don't know.
Hello, sir. Yes, sir. Okay, sir. Sir, it is visible. Hello, sir. Okay, sir. So, good evening, sir. My name is Mayuri Madvi and my roll number is 227318. So, my topic is screening of genomic library. So, first introduction. So, the term library can refer to a population of organisms, each of which carry a DNA molecule inserted into a cloning vector, or alternatively to the collection of all the clone vector molecules. And the DNA library is a collection of DNA fragments that have been cloned into vector to recognize and isolate desired DNA fragments. So there are two types of DNA libraries: genomic library, which are made from genomic DNA, and cDNA library made from copy of mRNA by reverse transcriptase. So what are genomic library? So genomic library is a collection of clones that when successfully made theoretically contain at least one copy of every DNA sequence in the genome. So genomic library have many uses in the molecular biology and in genomics. So remember that the key step in analysis of genomics is a breaking a genomic DNA into smaller, more easily manipulated fragments. A genomic library will contain these small fragments which are used in many types of genetic analysis. So how genomic library constructed? The first step is the extraction of genomic DNA, then second cut with the restriction enzymes. In third step, ligate the gene with the vector which cleave by the same enzyme. In the fourth step, transfer into bacterial cell which are able to undergo treatment term as a competent cells. So calcium fluoride causes DNA to precipitate on outside the wall of the bacterial cells. In the fifth step, amplifications. For example, E. coli cells are grown in agar medium containing amplicillin or tetracycline at 37 degrees Celsius. After the construction of genomic library, so the screening of genomic library performed. So it is screened to identify the gene of interest. One of the most common library screening technique is called colony hybridization. So in this process, library construction phage vector are used. Then the process of identification of gene of interest involved in this plaque hybridization. First, the colony hybridization. So, colony hybridization is screening of library with label probes or radioactive probes, etc. So, to identify a specific sequence of DNA, RNA, enzymes, protein, or antibodies. In the second, plaque hybridization. So, in that, uh, based on the hybridization of oligonucleotide probe to target DNA. So DNA is a transfer directly from the petri dish to the filter, so which is then incubated with label probes. So here we discuss the screening of genomic library made in a phage cloning vector through diagrammatically. So in a first step, this phage vector have small genomic library and the E. coli cells are infected with genomic library. Then the cells are plated as a lawn. In the second step, on next day, where plaques are produced. So in a third step, the membrane filter is placed on a plate. Phage particles which are present in the plaque stick to the membrane filter. So the filter is then processed to lyse any bacterial cells. So denature the DNA to single stranded, uh, then bind to that DNA to the filter. In a fourth step, so next the filter is placed in a heat sealable plastic bags and incubated with probe which have been labeled radioactively or non-radioactively. So here we use riboprobes which are made up of RNA and which are used as a radioactive probes. So since this can be created by in vitro transcription, so they can be labeled very simply. We just have to add radioactive NTP to the vitro transcription reaction to make radioactive riboprobes. So to use labeled DNA as a probe, the DNA is denatured by the boiling and then cooled quickly on ice to produce single-stranded molecules. So these label molecules are added to the membrane filter to which the denatured DNA or single-stranded DNA for colony has been bound. In the fifth steps after the hybridization, method was performed. The filter are washed to remove unbound probes and subjected to the detection of procedure for whether the probe was radioactive or non-radioactive. So this dark spot indicated the gene of interest uh, by by the method for radioactive probe by autoradiography and from non-radioactive probe through chemiluminescent or calorimetric detection. So here are I have explained this all step through diagrammatically. So now advantages of genomic library. So genomic library derived from eukaryotic organism are critical for studying the genomic sequence of particular gene of interest. So it is useful for prokaryotes with small genome to identify a clone encoding a specific gene of interest. So it helps researchers to explain more about an organism, genomic structure and function. It is also used to study genetic mutation. Pharmaceutically important gene can be identified by this method. Disadvantages of genomic libraries are It requires sophisticated software and vast amount of computing power and also process prone to error. So eukaryotic genome library with very large genome contain many DNA that do not code for proteins. So as well as non-coding DNA like repetitive DNA and regulatory region, making them less than ideal. Application of genomic library. Genomic library construction is the first step in any DNA sequencing project. So it helps to identification of the novel pharmaceutical important genes. It also helps to identification of new genes which were silent in the host. It helps in the understanding the complexity of genome, serving a source of genomic sequence for generation of transgenic animal through a genetic engineering. Study of function of regulatory sequence in vitro, study of genetic mutation in a cancer tissue. So here are my references. Thank you, sir. Thank you, sir. सर जीनोमिक लाइब्रेरी में इंट्रॉन्स प्रेजेंट होते हैं. हम्म, नहीं पता सर क्रोमोसोम का. सर इट इज इमेजिन टेक्निक होती है वो रेडियो यूज करते रेडियो एक्टिव सोर्स. इमेजिन इमेजिन इमेजिन. नो सर. थैंक थैंक यू सर.
सर व्हाट्सएप पे भेजा आपको पीपीटी. Hello everyone, good evening. My topic is phasmid with detailed explanation properties of one phasmid. So phasmid or phasmid are DNA-based cloning vector having both bacteriophages and plasmid traits. Along with the origin of plasmid replication, the vector carry an origin of replication obtained from bacteriophage. So replication is obtained from the bacteriophage. Plasmid, unlike plasmids, vary in having the ability to pack into the capsid of bacteriophage as they possess a genetic sequence signaling for packaging. They find application in different streams of biotechnology, which is called phage display. So as you can see in this diagram, poly ORI from plasmid uh, for antibiotic resistance is to identify recombinant plasmid, plasmid. It also has less gene multiple cloning side MCS. So in the pink region ORI origin of replication taken from bacteriophage and blue script to two type of ORI. So phasmid is ORI, AMP like operon, MCS, and phage means ORI. So the main advantage of the phasmid system is that it can be used to provide single or double standard material without recloning, without any recloning. The carrying capacity of plasmid is higher than phage vector, having higher efficiency of transformation. They are more stable than recombinant phage vector. They can be exploited to generate single-stranded DNA templates for sequencing. Example, PMBL8 was made by transferring into PUC8 a 1300 BP fragment of the M13 genome. This piece of M13 DNA contained the single sequence recognized by the enzyme that convert the normal DS M13 molecule into single-stranded DNA before secretion of new fast particles. All that is necessary is the E. coli cell used a host for a PEM-BL8 cloning experiment are subsequently infected with normal M13 to act as a helper phage, providing the necessary replicative enzyme and phage code protein. PEM-BL8 being derived from PUC8 has the polylinker cloning site within the LAGZ gene, so recombinant plaques can be identified into standard way on agar containing X curve. With PEM-BL8, a single standard version of cloned DNA fragment up to 10 kb in length can be obtained, greatly extending the range of M13 cloning weapon. Thank you, sir. I don't know, sir. Ampicillin, sir, ampicillin. Mm, don't know, sir. Mm, greenish, sir. Mhm. Yes, sir. Sir, white, maybe green, white. I'm confused, sir. Thank you, sir.
यस सर। सर मैंने आपको सेंड किया है। Good evening, everyone. My roll number is 227329. Topic is bacteriophage explanation and properties of one of the bacteriophage. Bacteriophage is a virus that infects, replicates within a bacterium derived from bacteria, and phagine means to devour. Single or it can be single or double stranded, surrounded by protein capsid. Genomes may encode as few as 4 genes and as many of hundreds of genes. So this is the diagram of bacteriophage. It composed of head, tail fibers, and the head has it is made up of protein coat, which is known as capsid, and it is encapsulated the genome. It consists of polyhedral head and there can be 2000 capsomers within the double stranded DNA enclosed within it, and it also contains the thread line structure, means whiskers, and means they are extended outwards from the cola region, and it also has spike proteins that penetrates and causes infection, and it has a tail, means, and the tail portion has six fibers holding the receptor to recognize the attachment site on the bacterial surface. It also has the inner hollow tube like structure which is surrounded by sheet with annular ring, and हाँ sir आगे. So phage are very simple in structure, consisting of DNA molecule carrying a number of genes surrounded by protective coat or capsid made up of protein molecule, and it undergoes within two life cycle, lytic cycle and lysogenic cycle. Next, why bacteriophage? They are act as the cloning vector. It can accept very large pieces of foreign DNA. Genetic engineers have constructed numerous derivatives of phage vector that contain only one or two sites for a variety of restriction enzyme. Phage that have a stuffer fragment are called substitution vector, means one can be substituted with something else. Next. So, derivative of phage have been developed as cloning vectors since the early days of gene technology. The phage derivatives are considered to be the most suitable cloning vehicle for cloning genomic eukaryotic DNA because of the following reasons. Because thousands of phage plaques pla- can be obtained in a single petri dish. Selection of DNA hybridization is possible. In vitro packaging is possible. Size selection of packaged DNA is possible. Millions of independently cloned virus particle can be substituted to form a gene library. So these are the example: lambda phage, M13 phage, T4 phage, T7, P1 phage. So, lambda phage, it is the coliphage, belongs to the Cyphovidae family, nearly 50 nanometer diameter, and it has the icosahedral head. It is flexible tubular protein tail. It connectors serve as the site of attachment of performed site of attachment. Cos sites on the end has the double stranded linear DNA molecule of MV31 Dalton. Because of the presence of cos sites, DNA adopts circular structure when injected into host cell. So lambda phage is non-contractile tail phage, meaning during an infection, e- even it cannot force its DNA through a bacterial cell membrane. It must instead use an existing pathway to invade the host cell, having envelope of the tip of the tail to interact with a specific pore to allow entry of its DNA to host. सर ये सब एग्जांपल्स ही है उसके. Thank you. Double stranded DNA. Adenovirus. Double stranded. डबल स्टैंडर्ड एडिनो वायरस और क्या बोलते हैं उसको, पॉक्स वायरस. सिंगल स्टैंडर्ड नहीं पता सर, उसका नाम नहीं याद आ रहा है। पैर 40 किलो बेस पर। 40। किसके कंपेयर में सर? मतलब वो मीन्स एक्सेल अगर पे अगर ग्रो करेंगे तो बहुत सारे ये कॉलोनीज मिलेंगे। वो एक है। और नहीं।
Yes, so yes. Okay, sir. May I start? Good evening, sir. Myself Aarti, and my topic for today's presentation is artificial chromosome with detailed explanation properties of one Y-acid, that is yeast artificial chromosome. So, firstly, artificial chromosomes are cloning vectors that can accommodate very large pieces of DNA, producing recombinant DNA molecules resembling small chromosomes. Artificial chromosomes are useful in genomic applications as we can use them to study large segments of chromosomes. Also, they can contain entire genome in manageable number of clones. Example of artificial chromosomes are bacterial artificial chromosomes, human artificial chromosomes, which comes under MAL, that is mammalian artificial chromosome, and yeast artificial chromosomes. So, I'm going to focus on yeast artificial chromosomes. So, yeast artificial chromosome is a human engineered DNA molecule used to clone DNA sequences in yeast cells. Yaks are often used in connection with the mapping and sequencing of genomes. Segments of an organism DNA up to 1 million base pairs in length can be inserted into yaks. The yeast artificial chromosomes with their inserted DNA are then taken up by yeast cells. As the yeast cells grow and divide, they amplify the yak DNA, which can be isolated and used for DNA mapping and sequencing. Uh, so basically, yak is a kind of vector which is actually originated from eukaryotic origin. It is artificial chromosome for yeast, as names suggest, yeast artificial chromosome. It is linear vector, but it can be made circular. And components of yaks are telomere, a yeast telomere at each end, then centromere, sequence allowing regulated segregation during mitosis, ARS, that is autonomously replicating sequence allows the vector to replicate in each cell. Each selectable markers such as TRP1, that is a tryptophan, and URA3, and bacterial selectable markers are also present. So construction of YAP. YAP is built using an initial circular DNA branches, which is typically connected to linear DNA molecule using restriction of the DNA ligase and then used to ligate a DNA sequence or gene to insert it into the linearized DNA, forming a single large circular piece of DNA. So this is a diagram I'm going to explain. Yak's particular vector can be maintained in bacteria as well as it can be maintained in the yeast. Host cell is also yeast, but it can be maintained in that only of replication with bacteria. And how vector looks like. This is this is the circular structure we have, and we have telomeres in it, and also we have centromeres, and in between we have restriction site. We have restriction site, that is BAM H1 site between two telomeres, between two telomeres, and this is the and also we have selectable markers. We have R1 eco R1, and which is cleavage site, and selectable markers also, which are used to screen the recombinant plasmids from the non-recombinant ones. And thing is, if we treat the circular structure of this yeast artificial chromosome with BAM H1, it will cleave it between it between from center, so that it will generate telomeres at one side, at both the ends, and centromeres in between, uh, centromeres in the center, and and yeah, that's the centromeres in the center. The properties of yeast artificial chromosomes. One arm contains an autonomous replication sequence ARS, a centromer, and a selectable marker. The other arm contains a selectable marker, that is URA3, have high cloning capacity as its inserts can be up to 1000 kilobase in size. Yaks are often chimeric. Uses of yeast artificial chromosome. Yeast artificial vectors are DNA constructs that are used for cloning DNA in yeast. Yeast artificial chromosomes have become essential research tool as they enable large fragments of DNA to be cloned. Yeast artificial chromosomes are often used in connection with the mapping and sequencing of genome. Yeast artificial chromosomes are extremely popular for those trying to analyze the entire genome to identify essential mammalian chromosome sequences necessary for the future construction of specialized mammalian artificial chromosome. Yeast artificial chromosomes are also used in human genome project. And that's it. And the references are iGenetics. Thank you. The drawback of YAP is it is quite unstable than bacterial artificial. used for. Hello. Used for. Important. सॉरी या आप क्या था? फिर एक बार हाँ एक बार डोंट नो. याक में ओराई नहीं रहता. एक मिनट याक में याक में ओराई रहता है बट वो क्या बोलते हैं सर देखो ईस्ट यूकैरियोटिक ऑर्गेनिज्म है. ठीक है सैक्रोमाइसिस सीरीज में जो होता है अभी एंड जो ओराई जो रहेगा वो मतलब उसका क्या बोलते विल नॉट वर्क इन ईस्ट सेल इसीलिए पहले उसको बैक्टीरिया में डाल के रुकते हैं. तो ईस्ट में ARA सीक्वेंस रहता है वो फिर बैक्टीरिया में वर्क नहीं करेगा ऐसे. थैंक यू सर.
Yes, sir. अगर बोले पापा बस हो गया. पानी आ रहा है तुम्हारे आ रहा है. पानी आ रहा है इसमें बाउल मत है. अरे ये कटोरी में सालन भी दे दीजिए. कहाँ सालन में एकदम पतला. So now it is visual. Yes, good evening everyone. My name is Ishad Ali. My topic is DNA cloning basic step. Now what is the cloning? Cloning describes the process used to create excess genetic replica of another cell tissues or organ. The copy materials which has the same genetics makeup as the original is referred to as a clone. The most famous clone was the Scottish sheep and name is Dolly. There are three different types of the cloning. Gene cloning, which creates copies of the genes or segment of DNA. Second is the reproductive cloning, which is create copies of whole animals. And third is the therapeutic cloning, which creates the embryo stem cells. Researchers hope to use this cell to grow healthy tissues to replacing injury or disease tissue in the human body. And what is the DNA cloning? DNA cloning is the molecular biology technique that make many identical copies of pieces of DNA, such as the gene. In the typically cloning experiment, a target gene is inserted into the circular pieces of the DNA called as a plasmid. And what is the basic step in the DNA cloning? First is the isolate DNA from an organism. Cut the DNA into pieces with a restricted enzyme and an enzyme that recognize and cuts within a specific DNA sequences and insert each pieces individually into cloning vector cut with the same restriction enzyme to make recombinant DNA molecules. A DNA molecules constitute, co- constructed into vitro cloning sequences from two or more distinct DNA molecules. Introduce transform the recombinant DNA molecules into a host such as the E. coli replication of the recombinant DNA molecules. The process of molecular cloning occurs in the host cell, producing many identical copies called as a clone. As the host organism reproduce, the recombinant DNA molecules are passed on the all types of the progeny, giving rise to population of the cell carrying the cloning sequences. सर जो मैंने यहाँ पे बताया वही यहाँ पे डायग्राम में एक्सप्लेन किया हुआ है. And importance of the DNA cloning. DNA cloning can be used to make proteins such as the insulin with the biomedical techniques. It is used to develop recombinant versions of the non-functional gene to understand the functioning of the normal gene. This is the applied in gene therapies also. It helps to analyze the effect of mutation on a particular gene. There are differences. Thank you. Hello. Yes. Thank you.